Okay, so I would say we get started. Today is again another session of our clinical trial conference series, which is a joint venture between GLAMCON and NephCure. Today's session is on diabetic nephropathy, and we have the honor to have Dr. Vlado Perkovic, Dr. Katalin Shustak, and Dr. Sylvia Rosas with us. Dr. Vlado Perkovic is a Dean of Medicine and Scientia Professor at University of New South Wales, Sydney, and prior to this, he was the Executive Director of George Institute. He's a staff specialist in nephrology at the Royal North Shore Hospital. Dr. Perkovic's research focuses in clinical trials and epidemiology, particularly in preventing the progression of kidney disease. He leads several international clinical trials and played a central role in the development of an affordable dialysis system, which was a Eureka Prize finalist in 2017. He serves on the editorial board of New England Journal of Medicine and is associate editor at the Journal of American Society of Nephrology. Dr. Katalin Shustak is a physician scientist and a professor of medicine and genetics at Perelman School of Medicine, University of Pennsylvania. Her laboratory interest is in understanding the pathomechanism of chronic kidney disease development. Dr. Shustak has made discoveries fundamental toward defining critical genes, cell types, and mechanisms of chronic kidney disease. She was instrumental in defining genetic, epigenetic, and transcriptional changes in diseased human kidneys. She has identified novel kidney disease genes, and her lab was the first to map the kidney epigenome and catalog genotype-driven gene expression variations in human kidneys. Dr. Shustak generated the first unbiased comprehensive kidney cell type atlas using single-cell transcriptomics. She identified that specific renal endophenotypes are linked and likely caused by the dysfunction of specific cell types. Dr. Shustak's discoveries span genetics, genomics, epigenetics, molecular biology, physiology, and nephrology, and have enormous translational relevance and considerable therapeutic potential. Dr. Shustak has been the recipient of the 2011 Young Investigator Award of the American Society of Nephrology and the American Heart Association, one of the most prestigious awards given to researchers under the age of 41 in the field of nephrology. The session will be moderated by Dr. Sylvia Rosas. Dr. Rosas is an associate professor of medicine at Harvard Medical School and nephrologist at the Joslin Diabetes Center and the Bethesda Deaconess Hospital in Boston. She is the director of the Latino Clinic at the Joslin Diabetes Center. Previously, she completed her nephrology and epidemiology training at the University of Pennsylvania. Dr. Rosa's primary research focuses on the epidemiology of metabolic and cardiovascular disease complication in the setting of chronic kidney disease. She is a PI for the NIDDK APOL1 long-term outcome in kidney transplantation network and the Kidney Precision Medicine Project. She is an investigator for the NIDDK Preventing Early Renal Function Loss multicenter study evaluating the efficacy of the urate-lowering drug allopurinol as compared to placebo in reducing kidney function loss among subjects with type 1 diabetes. Her research has been funded by the National Institute of Health and the Veterans Health Administration. Also to note that this session is basically made possible under the support and collaboration with the NephCure Foundation. And we are grateful to the Kidney Health Initiative and the American Society of Nephrology for helping us promote and raise awareness about this series and organize this event with us. Special thanks to Lauren Lee, Chief Research Officer, NephCure Kidney International, and the co-organizers, Dr. Kirk Campbell and Dr. Sunil Udani. And so without much delay, I would say we get started and give the first one to Dr. Perkovic.
Thank you very much, Ali, and thanks for the opportunity to speak to you today. I'm going to talk briefly about a little bit of where we've come from and where we are now, I think, and start looking forward in terms of the management of diabetic kidney disease. Go to the next slide. If I can drive this. These are my disclosures, and during this, I'm really just going to touch at a very high level over, I think, some of the key concepts that have arisen out of some of the data that we're seeing. We go to the next slide. This is the reason that we've been able to, I think, move forward fairly dramatically in diabetic kidney disease is that we have a condition which is now the leading cause of kidney failure in the US, and that's been important in being able to allow us to convince people to invest in the area of diabetic kidney disease. And as a result, we've had a flurry of trials that have helped to guide treatment. On the next slide, we'll see the other important factor is that the issue is not just the risk of kidney failure, but we've also had to be cognizant of the risk of cardiovascular events, which is, as we know already, substantially increased in people with kidney disease, but it's particularly high in people with diabetes and kidney disease, as shown in the figure on the slide. We go to the next slide. This sort of is a summary of where we were up until a few years ago. I call this a mega meta-analysis. This is a figure that we created for a review article that summarizes 5 separate systematic reviews, looking at the effects of various cardiovascular interventions in people with chronic kidney disease. At the top, we see that, if we could just go back, blood pressure lowering appears to be effective overall, 17% reduction in risk for 5 millimeter lower blood pressure. Lipid lowering overall is effective. Some evidence for fibrates having a modest effect, and some evidence for aspirin having a moderate effect, all in people with chronic kidney disease for the prevention of cardiovascular events. But if we look at this data in a little bit more detail on the next slide, what we see is that at least for lipid lowering, there's some reasonably strong evidence now that things are not the same at all different levels of kidney function, that people with earlier stages of CKD may have greater relative risk reductions for cardiovascular events, at least in than people with more advanced kidney disease. And those on dialysis or stage 5, it really wasn't possible to demonstrate a clear benefit overall. If we go to the next slide. So we started off really by looking at secondary analyses of trials done in the general population, subgroup analyses, a focus on cardiovascular disease. And then, of course, Renal and IDNT, both published in 2001, were the first treatment identified to prevent kidney failure in people with type 2 diabetes. And this was a huge step forward for us, risk reductions of only 16 to 20%, which we, until perhaps a year or two ago, thought were very large. Don't look so large now in the context, but also with a very high residual risk of disease. Even in the people randomized to angiotensin receptor blockers, the rates of kidney failure were still very, very high, highlighting the need for additional treatments. We move to the next slide. Subsequent to that, there were a series of trials of a whole range of different therapies, and there were some important lessons out of these. One was that things that appeared to be effective in animals or even in early phase human studies didn't always turn out to be effective, and many of these treatments actually turned out to have no benefit. And the second key lesson from all of these trials, for me at least, is that we must be alert to the risk of adverse effects that perhaps are unexpected related to some of these treatments. And we saw that, for example, with hyperkalemia and dual RAS blockade, with fluid retention, with endothelin antagonist, and heart failure with bardoxolone. So demonstrating efficacy is crucial, but we also must keep an eye on the risk of adverse events and make sure we consider those appropriately. We move to the next slide. This, of course, is then the data from the Credence trial, which we reported in April last year in Melbourne, where we compared canagliflozin to placebo on the risk of a composite renal and cardiovascular outcome, and showed that canagliflozin reduced the risk of end-stage kidney disease, doubling of creatinine or renal cardiovascular death by 30% compared to placebo. If we move to the next slide, we see that these results really were very consistent for a whole range of renal outcomes, regardless of how we define them. And again, another key lesson here is we must give really quite a lot of thought to which of the key outcomes we might choose to use, which combination of the various potential outcomes we look at, because this has substantial implications for study design and size. We move to the next slide. And just if we can click through this in the interests of time, this is what happens to EGFR. Again, we see this acute effect, which is similar to RAS blockade, and then a stabilization with a 2.7 mil per minute per year difference, or a 60% reduction in the rate of loss of EGFR after that acute effect, translating into substantial benefits. If we keep going. On the next slide, We then see a summary meta-analysis that we published in Lancet Diabetes and Endocrinology subsequently that pooled the effects on the hardest renal outcome, dialysis, transplantation, or death due to kidney failure across Credence and the 3 completed cardiovascular outcome programs. And what we see again is a very clear-cut 1/3 reduction in the risk of this outcome. So I think there's no uncertainty now. It's very clear that SGLT2 inhibitors prevent kidney failure. These results would suggest that the benefits may be a class effect, but of course, we await the outcomes of ongoing trials to confirm whether or not that's the case, which I think it will be, but also help us understand whether the magnitude of effect with different agents is the same. We move to the next slide. The other things we've learned then from, particularly from meta-analyses of these different trials, is that the benefits appear to be relatively consistent at different levels of albuminuria. You see at the bottom the macroalbuminuric population, the microalbuminuric population in the middle, and then the normoalbuminuric population at the top. And even in people with normal levels of albuminuria, we see a substantial, almost halving in the risk of the composite renal outcome. And that may have implications for mechanism. If we go to the next slide, we also see similar results, although evidence of perhaps some attenuation of the magnitude of effect at different levels of EGFR, with clear benefits even in the subgroup with an EGFR below 45 and at all EGFR levels above that. But a p-value for trend of 0.07, suggesting perhaps the relative risk reduction gets a little bit weaker, but it's important to note that the people with lower EGFR are at higher absolute risk, so the absolute risk reductions are even greater. We move to the next slide. The other thing that we're able to do in Credence is look at the people who were enrolled based on an EGFR between 30 and 90 into the screening period. And then what happened was 4 to 8 weeks later, people had an additional EGFR taken at randomization, but they were already randomized by the time that EGFR was done. So that EGFR was not considered in terms of the eligibility, but it meant that there was a couple of hundred people who had an EGFR that had already fallen below 30, and we're able to look at what happened to kidney function in those patients. And even in the people who had an EGFR below 30, mostly between 20 and 30, we saw a significant slowing in the rate of loss of EGFR decline, 2.5 mils versus 2.5 mil per minute difference, even in people who had an EGFR below 30 at randomization, suggesting that the benefits of these agents may extend even below 30. We go to the next slide. Here we see an additional subgroup analysis that looked at effects by baseline HbA1c, and even in the participants who had a baseline HbA1c less than 7, in whom the A1c effect was really minimal, barely detectable, we see a clear reduction in the risk of the renal outcomes. No evidence that it's in any way dependent on the HbA1c, and highlighting the likely glucose-independent mechanism of benefit of these agents. We go to the next slide. The same is true really for cardiovascular outcomes. I won't go through this. This is a recently published meta-analysis that shows, again, overall benefits for a whole range of cardiovascular outcomes. There's some evidence that, in fact, the benefits on MACE may be slightly greater in people with an EGFR below 60. And the individual outcome that seems to drive this is stroke, where there appears to be a benefit for people with kidney disease that's not seen in people with preserved kidney function. It's not clear whether this is a real effect or whether it's due to chance or what the mechanism is, but it's an interesting observation. If we go to the next slide, we then see data also recently published from the DAPA-HF study, and this study was important. It enrolled people with heart failure and looked at the effects on cardiovascular death, heart failure hospitalization, or a urgent heart failure visit. But the key thing was that it included people who had diabetes, but also people who did not have diabetes. And the benefits, a 26% risk reduction overall, were pretty much identical in people with and without diabetes, again highlighting the likely glucose-independent mechanism of benefit of this class of agents. We move to the next slide. This just really points out that so far we have the results of 5 SGLT2 inhibitor outcome trials, but we're not even halfway into the SGLT2 inhibitor journey, with a large number of trials still underway and due to report over coming years. And perhaps the most exciting of those for us is DAPA-CKD, which, as I understand it, may report towards the end of this year. And that's, again, important because it includes people with non-diabetic CKD. So we'll also learn whether the kidney benefits are equivalent in people without kidney disease. We go to the next slide. One of the key things to note is that we're also getting evidence of treatments that don't work. This is the Carmelina trial, 7,000 people enriched for the presence of kidney disease and quite a lot of renal outcome events, 633 primary renal outcomes, but very clearly no benefit with linagliptin compared to placebo for that renal outcome. A greater A1C effect than we saw with Credence. So highlighting again the fact that this benefit is not due to glucose lowering. It's not seen with necessarily all classes of glucose lowering agents. And in fact, that DPP-4 inhibitors don't really appear to have any benefit for hard renal outcomes. On the next slide, we see, and I'll just get you to click that again, some data from LEADER and SUSTAIN-6 looking now at the GLP-1 receptor agonists with the effects on a whole range of different outcomes, looking at pool data at the top there. Some suggestion that some of those outcomes are 40 or 50% decline in eGFR. There may be a modest benefit, 15 to 20% with GLP-1 receptor agonists in a pooled analysis of these studies. If we click again, we'll see the same data broken down by kidney function. And interestingly, for this class of agents, from the LEADER and SUSTAIN-6 study, there is a suggestion, which was statistically significant in terms of the heterogeneity, the benefits may actually be substantially greater in people with reduced kidney function, and point estimates of benefit ranging from 35 to 45% for the various outcomes in people with an eGFR of 30 to 60 and or albuminuria. So this is promising data, and if we click to the next slide, we'll see that this is matched, that it's also important to note that the effects in these trials on some of the outcomes, and here we see albuminuria data, varied by drug. So liraglutide 1.8 milligrams and semaglutide 0.5 milligrams both appeared to lower albuminuria by about 20%, but oral semaglutide at a higher dose, 1 milligram, lowered albuminuria by a third. And again, these were significantly different from each other. We saw similar differences on eGFR slope and other outcomes, suggesting that the choice of drug may also be important when we start looking at the GLP-1 receptor agonists. And on the next slide, we see that hopefully we'll learn more about this over the next couple of years. The FLOW trial is a specific renal outcome study with semaglutide 1 milligram, comparing it to placebo in people with type 2 diabetes and kidney disease. We go to the next slide. The other recent trial that obviously has been important is the SONAR trial, which looked at atrasentan compared to placebo. There were a number of interesting design elements that had unintended consequences in terms of event rates, and also demonstrated that it's very hard for us to predict people who truly have reductions in albuminuria in response to therapy. And I think there's some important lessons there for future trials. But even in this trial, we saw a 1/3 reduction in kidney failure events amongst responders, with similar suggestion of benefit amongst non-responders. If we go to the next slide. To summarize, we know that our patients with kidney disease are at high risk. I think the trials that we've just seen tell us that we can do this. We can identify in a robust way treatments that work to prevent kidney failure and also prevent some of the other comorbidities in people with diabetes. And SGLT2 inhibitors clearly prevent both kidney failure as well as cardiovascular events. And they also appear to at least prevent heart failure in non-diabetic patients as well. So this is an exciting area of future study. DPP-4 inhibitors don't work. GLP-1 receptor agonists may have some benefit, perhaps particularly in people with kidney disease. And endothelin antagonists would appear to have a potential role in the future and are now being studied in glomerular diseases. As we learn about the benefits of drugs, start to realize that there's a big gap in what we do in terms of getting these treatments to the people who will benefit from them and how they might work in combination. So these are going to be important areas for future study. Thanks very much.
Excellent, Dr. Perkowitz, for that presentation. If you have a question, you can start writing your questions in the chat and we will answer that later. But now we're going to listen to Dr. Susak's presentation, who will give us a different perspective, sort of trying to move more into the basic science world and how that can help us with clinical work.
Well, thank you so much for inviting me to this chat. This is my first time to work, and I would like to disclose that I am in heart a real basic scientist, a mouse biologist, even though I'm a practicing nephrologist. So I do have a slight different view about things. So we can go to the next one. So when we consider what is next available for diabetic kidney disease therapeutics, I put this slide together about a recent review in Nature Reviews of Nephrology by Joachim Andes, that what are the underlying mechanism of disease. There are clear hemodynamic and metabolic effects that lead to cellular and tissue injury. And I put the SGLT2 inhibitors and maybe the GLP-1s into this category, although I will be interested to hear Dr. Perkowicz's opinion, whether he feels that this is really the right grouping. Then there are several other pathways that have a well-known contribution for disease development. The tubules that are essentially have a hyperreabsorptive stress for hyperglycemia. They undergo hypertrophy initially, and then later they undergo atrophy. They undergo tubulointerstitial fibrosis. The endothelial cells, which play a very important role in disease development. After all, kidney disease is called the microvascular complication of diabetes. The glycocalyx has received quite significant attention, loss of fenestration, but overall at the later stages, there is really a clear loss of the microvasculature, which probably an important contributor to glomerulosclerosis and fibrosis development as well. And then also the podocytes seem to be very important for disease development. There is ample of data indicating that the hyperfiltration and the enlargement of the glomerulus associated with podocyte hypertrophy, enlargement of the podocytes at early stages of disease, but later stages the podocytes are lost, which is important for glomerulosclerosis. So using this as an overall, I tried to put together several potential pathways that may be important for future targeting, and I will illustrate it in the following slides. So the next slide starts with, and I think it has another side so you can animate it in. Let me just let this go. So it will tell about the mineralocorticoid receptors, and then we know that they have been tried. Spironolactone and several others seem to actually have a quite beneficial effect in different conditions, although the clinical trials, in my understanding, haven't really panned out for this class of drugs. They seem to interfere with multiple things, including the reactive oxygen species generation, NO and endothelial function, different pathways of fibrosis, of inflammation and cytokine, and they influence several things, including glomerular hypertrophy, glomerulosclerosis, and injury. Although we know, and I think there is an animation on this side as well, that spironolactones are associated with multiple side effects. Hyperkalemia is one that we observe quite often, and then patients with diabetes often tend to have hyperkalemia on their own. So maybe the limitation of this class of drugs have been by their side effects as well. So the next slide will show you that there is a new compound that has been developed, is firenone, which is a novel non-steroidal MR antagonist, which has a massive selectivity for MR, 100-fold, It also has higher potency and a decent half-life, and it would essentially act on the mineralocorticoid receptor system. Next slide. Next slide. And so there is an early clinical study performed by fluranilon in patients with diabetes and albuminuria. It was an early phase 2 study, but what you see is that the drug actually seemed to have a very significant effect on albuminuria, which is usually the outcome of phase 2 studies, indicating that there is a positive signal for this specific drug. The next slide will show that based on that, 2 studies were actually initiated, the Fidelio and the Figaro, which is going to be essentially assessing the effect of fluranilon, this new class of drug, in reducing kidney failure and disease progression in patients with diabetic kidney disease. The next one. And this is the study design, and it is my understanding that the study will actually complete sometime this year, or maybe early spring or early summer. This is, this is an informed consent. There is a randomization. There are 2 doses of drugs depending on the GFR, and they will monitor potassium as a side effect and GFR, and that will be read out as a study termination. So these patients will be on ACE and ARB or a combination of both depending on patient guidelines. So this will be interesting to note what's going to be the outcome of this new drugs trial. The next one. So I put the MR antagonist over here, which will act through also hemodynamic and hypertrophic stress. And then the third class of drug is also being studied. That's called an ASK inhibitor, which the mechanism of action for this will be reducing the inflammation, scarring, and tubular interstitial fibrosis. The next slide will show you the paper, which was actually an animal model study, which have shown the benefit of this particular inhibitor using different models of kidney disease. This is actually a mouse model of diabetes, and what you see is that these animals have reduced GFR from baseline to 18 weeks, and then the drug, the GS, actually has an important protective effect on both on the GFR, has a significant protection of albuminuria, and then the lower panels also show that the drug showed a very promising effect on protecting the animals from fibrosis development. And the authors actually tested multiple other mouse models, and the effects seem to be quite consistent. The next slide shows that based on these animal studies, the drug has been tested also in patients, 333 patients with type 2 diabetes, with diabetic nephropathy. The mean GFR was relatively low, I would say, 31. And then they were either on placebo or different doses of drugs they were assigned. And the primary analysis was different in the eGFR, and there was an exploratory analysis in the eGFR slope. Interestingly, this study did not meet its primary endpoint, but the post-hoc analysis suggested a very positive signal. And then so it's my understanding that the drug has been taken to a much larger phase 2 studies through the periodic hypothesis that it's actually protective from fibrosis development. So we will hear about this several years from now. So what are the next steps? So these are obviously, we went from studies that have already demonstrated benefit to studies that seem to have substantial promise based on phase 2 studies that are ongoing phase 3. But really the questions are like, what are the next steps? And I think I completely agree with Vlado's point that several of the pure mouse model based approaches are probably not fully translatable to clinical trials. So patient-centric approaches, I think, are critical for demonstrating target engagement and for new drugs. Unbiased approaches are definitely much more affordable than they have been before, and obviously they provide a completely unbiased view. And the other important aspect will be that instead of using the same drug for multiple patients, most likely patients with diabetes are also heterogeneous. Some patients have, rapidly progressive GFR decline. Some patients have albuminuria, but it seems that there is a subgroup of that patients who don't have albuminuria. So precision medicine-based approaches will likely be important for the future. So the next step actually shows some of our very own studies, which I lead, called Trident, Transformative Research in Diabetic Kidney Disease. It's a phase 0 major academic industry collaboration, which has UPenn as a coordinating site, but actually it has 21 recruiting sites. I would have to update that. And it has pharma and actually I would have to update this slide as well, but other partners such as the Juvenile Diabetes Foundation that are involved in this very complex relationship. So what is the goal of this? That essentially we use patient samples and generate a testable hypothesis for future interventional trials that could essentially provide the basis of future transformative therapies. We would like to identify pathways that are associated with rapid progression. And then we will do that via a multi-omics characterization of patients, and that include genetic studies, genomic studies, histological studies, epigenetic studies, and clinical and biomarker profiles of roughly 200 to 250 patients that are followed. And then in these cases, what is very important, I think, in the field of diabetic kidney disease, that the diagnosis is ascertained by a kidney biopsy. So we will have tissue samples and omics profiling from patients' kidneys as well. And then we will be able to associate the changes with the biomarkers and the progression. The next slide. This shows the study design. So essentially everybody who has diabetes is eligible. There is a pre-screening, a kidney biopsy, which is usually prescribed by the treating physician, are important because we will usually collect a research core from everybody who agrees and signs an informed consent. The tissue undergoes this complex multi-omic analysis, which includes RNA sequencing, epigenetic analysis of the kidney, of the different compartments, and then a cell type, single cell level. We look at the pathology. We have a digital pathology repository. We look at the light microscopy, the electron microscopy. And then the diagnosis of diabetic kidney disease is actually ascertained based on the classic criteria of having GBM thickening. And then those patients who do have diabetic kidney disease are enrolled and followed, and the outcome of the kidney function decline is ascertained over a period of 4 measurements of GFR. We also collect genetic information, exome sequencing, biomarker, and even a urinary single cell sequencing is involved. So hopefully the study will stop enrollment sometime next year, and then we will be able to report all this comprehensive analysis and hope to identify new pathways for progression and maybe validate of many other existing pathways that have been proposed. Next slide. So I will have just a few teasers over here. What have we learned from such unbiased approaches? So we have actually, this is a publication from my very own group where we looked at using about 100 kidney samples from patients with diabetes and non-diabetes but varying degree of kidney function decline. And we did this unbiased transcriptomic analysis, and we correlated changes that we observe in kidney structure, such as fibrosis and glomerulosclerosis, and kidney function, such as GFR. Interestingly, what we found is that the gene expression changes correlate much better with structural changes such as fibrosis and glomerulosclerosis than GFR. GFR is really a functional readout of the kidney. And then there is some correlation with fibrosis, but not a complete correlation. And when we looked at what are the key pathways that are differentially regulated in these patients, we highlighted two key pathways. One is activation of the immune system, different immune pathway activation. And the other one is the loss and decrease and imperfect function of metabolism in patient samples. The next one, and if you could animate this, there is actually quite significant amount of data accumulating that there is a very significant metabolic dysfunction that drives chronic kidney disease development. One more. In a simplistic way, this is a study from my group, but there are several other publications of this. The renal tubule cells that essentially make up the largest mass in the kidney preferentially and only specifically use fat as their energy source. They are highly energy dependent because they have the sodium potassium ATPase as their basal membrane, and they perform actually solid carriers. They have the highest mitochondrial content in the body, so they need a huge amount of ATP for the kidney to work. And in disease conditions, then the mitochondria is not working. There is a defect in fatty acid oxidation. There is a reduced ATP, and that's a very significant feature of chronic kidney disease, including diabetic kidney disease. The next slide will show you that this actually provides this paradigm of metabolic dysregulation actually provides ample of opportunities for intervention. The fibrates were mentioned by Vlado, which is actually specifically regulate this transcription factor PPAR alpha, and then actually there specifically increase fatty acid oxidation, mitochondrial oxidative phosphorylation. In animal models, their effect size is really, really large, but if you look at patient data, they still remain significant in patient as well. There are several other drugs and pathways that essentially act upstream of that. Some of them are regulators of the AMPK kinase, ICAR, and actually metformin is also here. There are trials with microRNAs like the microRNA 21, which is also an upstream regulator of metabolism and fatty acid oxidation. And there are a couple of other fancy ones, FXR, LKB1, or TAG1. These have also been published, and in mouse models all seem to regulate this specific pathway, and in animal models they show quite significant effect. And I would also like to say that the SGLT2 inhibitors may actually fit into this paradigm because they actually reduce the workload of the proximal tubules. So if you don't use, if you don't have to reabsorb so much salt and glucose, then you have less of a demand for fatty acid oxidation and mitochondrial beta oxidation. The next one, here is one of the energy requiring pathway over here. Sorry, it has many animations that can be targeted over there. One very interesting aspect of this metabolism is actually that it generates this intermediate metabolites, the NAD, which is critical for generating ATP substrate for fuel metabolism. And then what is interesting is that the NAD is actually a natural metabolite of vitamin B3, and it can be augmented in patients. And then it actually, our chair, Poyan Mer, has actually showed that it actually has a significant effect. It was in the setting of an acute kidney injury, so obviously administering the drug and the outcome is much simpler than in chronic kidney disease, but the general idea is very similar, which essentially augmenting the metabolism of the tubule cells in the context of AKI or in CKD would be beneficial. Next one. Another interesting aspect of the metabolism is something that my lab worked very recently, which is actually quite interesting to us. So the mitochondria is actually an archaic organelle, right, that has been endocytosed by our eukaryotic cells earlier. So the mitochondria has an ancient circular DNA. So, I mean, the mitochondrial metabolism doesn't really work well. This mitochondrial DNA can actually release and get into the cytoplasm of the tubule cells. And then the tubule cells obviously recognize this ancient mitochondrial DNA as a foreign DNA. So activate the immune system, and there is a specific immune cell pathway, the SIGA-STING pathway, that actually recognizes foreign DNA inside the cells, leading to this cytokine activation and inflammation, indicating that this metabolic dysregulation actually can directly drive inflammation and fibrosis development in the kidney. And then we show this very, very nicely in the mouse model setting, but there is actually significant correlate in patient samples as well. Interestingly enough, this pathway is actually targetable, and there are new inhibitors have been developed for this SIGA-STING pathway. The next slide shows the inflammation that has been noted in the setting of diabetes by multiple investigators in the past. This is a review that probably not the recent one, but there are several of these cytokines have been tried and targeted in smaller phase 1 and phase 2 studies in the context of diabetes. Some had some effect, that includes the JAK inhibitors, and some of the CCR2 inhibitors had effect as well, but interestingly enough, none of them has been taken to a phase 3 study. And it will be interesting to know whether they actually have an effect on disease development. And the final slide will show you that, so we filled in multiple gaps in here that are potentially can be targeted for patients in diabetic kidney disease. We haven't filled up the purple one, which is the glomerular one, and that's what it seems to be the most specific for diabetic kidney disease, the glomerular sclerosis, the GBM thickening, which is really clearly linked to podocytes. And then the last slide of mine will show you there are several opportunities to potentially actually interfere with the podocytes or with the endothelial cells. One of them was mentioned earlier by Dr. Perkovich, the endothelial antagonism and the anthracentan. But there are other ways to potentially target the podocytes, and there is actually quite tremendous interest around that by big pharma or biotech, generate podocyte-specific drugs, which likely could work in the setting of diabetes, but maybe also important for other podocyte diseases, such as FSGS or maybe minimal change disease. And there is tremendous interest over there, and there are several pathways here that are highlighted that could be potentially targeted. And then this will be my summary slides. So there are 2 phase 3 studies that are ongoing with 2 different targets, the mineralocorticoid receptor antagonist or the ASK1, which targets apoptosis and fibrosis. I think we are in the area where we really have to pay a lot of attention for using multi-omic approaches. And even though I am a card-carrying mouse biologist, but I do feel patient characterization is critically important for the next phase of target identification, target engagement, and probably even early phase clinical trials of looking at efficiency. There are 2 key pathways that these unbiased approaches already highlighted. One of them is that dysregulated metabolism in the context of diabetic kidney disease and CKD. They could be potentially targeted, some of them by the fibrates, by microRNA, or by NAT supplementation that have shown promise in early clinical studies. The metabolic dysregulation lead to inflammation, which have been tried and then had shown some promise in phase 2 studies, but haven't been taken to a phase 3 clinical trial. But it would be very, very interesting to know whether they have an effect in diabetic kidney disease development. Obviously, originally we thought diabetic kidney disease is a non-inflammatory kidney disease. but we definitely see activation of cytokines and immune cells. Question would be is whether we should take a targeted approach. Only some patients should be treated with this drug. And then I would like to highlight that the podocytes and endothelial targeting, obviously endothelial targeting may be closer than podocyte targeting, is of a great interest for diabetic kidney disease drug development, but also probably for other diseases such as other glomerular diseases, FSGS, and minimal change, where this could have important role. So that's what I have, and thank you so much.
So I was saying that these were two fabulous presentations, and now we're going to start our question and answer session. We have our first question there was a question about whether Trident was recruiting both type 1 and type 2, or mostly type 2 patients. You mentioned the JDRF, so I was assuming yes, but.
Yes, so we just actually partnered with JDRF, and then we are enrolling. We always enroll type 1 patients, but the patients with type 1 diabetes are specifically supported by JDRF. To be fair, Trident already, we have close to 250 patients, maybe 200 with biopsies, and out of that, 10% of the patients actually have type 1 diabetes, so we get a good subgroup for type 1 diabetes as well. It could be very interesting because they seem to have their disease may be less multifactorial in terms of having obesity, hypertension, and so on. Maybe they allow us to interestingly, identify the pathways better than in type 2 diabetes, which is a true multifactorial disease of, hypertension, obesity, and metabolism as well.
Okay. So while we get other questions from the audience, I wanted to ask maybe both of you, but we'll start with Dr. Perkovich. It took a long time to get ACE inhibitors and ARBs to be used clinically in the community, and even nowadays it's still part of our goals to put everybody with that has an indication to put them on ACE and ARB. Now we have this also very effective medication. How are we going to get the community, the nephrology community, to prescribe it?
Yeah, it's a big problem, and it's a big challenge for us now, I think, with, if you think about all the people out there with diabetic kidney disease who might benefit from RAS blockade and now from SGLT2 inhibition, in many countries, half or more of patients still don't know they have diabetes, including in many high-income countries. Then of those who have diabetes, we still hear regularly that perhaps not a minority, but a significant number never get their kidney function, or rarely get their kidney function tested, and don't have albuminuria measures. So again, not only might they not have their diabetes identified, even if they're known to have diabetes, they might not have high renal risk identified, and then they need to be put onto these treatments. So there's multiple barriers to getting people onto the right treatment. And I think, in many ways, our current sort of healthcare paradigm doesn't work for protective therapies like this. And maybe we have to explore some different approaches. There are lots of studies even today that show that of people with diabetic kidney disease, a minority are on RAS blockade, let alone SGLT2 inhibitors on top. So we've got some real work to do with regards to implementation of proven therapy.
Can I just ask a question? So when I go and see the patients, I see the fellows have always some sort of a resistance of starting the SGLT2 inhibitors. But really, do you see any real contraindication for starting somebody from an SGLT2 inhibitors? They always ask, well, but maybe the glucose will go down, or the patient is in an R. But when I read your studies, it doesn't seem that low GFR is a real contraindication, or having or not having proteinuria. So I don't really see a clear contraindication. And I think if that's the case, maybe we should advertise it more broadly, because that's one of the limitations I see when I see patients in my clinic.
Yeah, I think you're right. I think there's a fear. I mean, they do need a bit of management. Some people have blood pressure, which might drop a bit. The glucose-lowering effects in people with kidney disease are generally not relevant. So I think there's a fear of those, and we've got to get used to using these drugs. When I completed my training, we used to admit people overnight to give them a dose of ACE inhibitors, and now we very quickly learned that was completely unnecessary. And I think a lot of the fears around SGLT2 inhibitors are similar. It's just that people aren't used to using them. I think the more we use them, the more comfortable we'll become, and that sort of fear will go away. And that's one of the big challenges, because even in endocrinology, there are drugs which are much easier to use. DPP-4 inhibitors have a placebo-like tolerability profile, but they also have a placebo-like efficacy profile. We're really wasting our patients' time and money with these sort of drugs.
Yeah. So we have a few more questions about SGLT2. The first one, is there any specific management you do for diuretics or other antihypertensives? Or if you start SGLT2, do you have to change the dosing of the other glucose-lowering medications?
Yeah, these are important questions. And one of the things that we did with the Credence trial in particular was we tried to make it as simple as we could. And I'd suggest that people follow that approach. What we did in Credence is we brought people back a few weeks after starting the drug. We didn't adjust any of those drugs in advance. Now, if someone's looking volume deplete or getting dizzy when they stand up and they're on diuretics, it would be sensible anyway to think about what the dose of diuretics should be. And it's true that people with reduced kidney function are potentially at higher risk of volume depletion. But I think the key thing is to start the drug and bring patients back a few weeks later and make adjustments then. If someone, in the rare situation where someone is on insulin or related agents and getting frequent hypoglycemia, then I think it is worth being careful. But that's not true for most of the patients that we're talking about.
Yeah. Okay, thank you for those questions. This is a question for Dr. Sestak. Do you see us in the future perhaps using some biomarker that you may find in your studies, or to sort of make trials that are not as large or that are shorter, that maybe enrich with people that really are going to develop diabetic kidney disease? You think that's coming?
Yeah, absolutely. I think that's one of the key goals of our study. And then obviously we feel that at least in one trial we would need to get kidney biopsies, but I hope that this will be maybe the last study to use kidney biopsies, and maybe non-invasive biomarkers can be used the same way as the biopsy genetic markers and blood biomarkers. That's one of our main goals. We're not 100% sure. As I had disclosed, actually the structural changes and the functional changes do correlate, but do not fully correlate. So we see quite significant discrepancy between kidney function and kidney structure changes. And then hopefully we will be able to find a biomarker that better represent the kidney structural changes. There's several other questions we got on the chat for Trident. Should I answer them?
Sure. What is?
So one question is how difficult to recruit for Trident since it's a biopsy required, which is a very important and interesting question. So when we started this was several years ago, I wanted to make sure that the risk of the kidney biopsy is really lies on the physicians and patient relationship. So the biopsy is actually ordered by the treating physician, and we only use an additional research core from an already indicated biopsy. Interestingly, this shows quite significant regional differences. Some centers recruit better than other centers, some things that we did not expect. Roughly about 50 to 60% of patients agree for an extra research core, according to our analysis.
Okay.
And then we were asked whether we have a European center, and we don't have a European site. This is only in the United States.
Okay, excellent. There was a question also about the use of SGLT2 inhibitors in type 1 diabetes. My understanding is that there's an ongoing trial, but what is Dr. Perkovich?
That's right. I think, there's quite a bit of evidence now suggesting that these drugs can be useful for glucose control in type 1 diabetes. There are studies looking at the cardiovascular and renal effects of SGLT2 inhibitors in type 1 diabetes. But the data also suggests that these people are at particularly high risk of diabetic ketoacidosis and actually have a very high risk of this condition. So I think the jury's out. I think there's no reason to think that they won't be beneficial in that population. The question will be about the balance between the benefits and the risks. And I think it's really important that we have the data from the proper randomized controlled trials before we go using these agents widely.
Absolutely.
So this is Ali. I had a question, and can I ask? The SGLT2 inhibitors is like a lot of questions come from that, and has taken our community by big excitement, and completely, I would say, surprisingly, the results were. I think, like as I understand, many in the nephrology community earlier were very skeptical to actually develop that for or bring it forward as for renal indication, and it was primarily for a diabetic endocrine indication, and then actually there was a big concern that it could have adverse renal side effects. So the fact that they really carried on and showed that degree of efficacy took us by surprise. But that was not only the only surprise. The other surprise was that it did protect the kidneys without really affecting glycemic control. So two major surprises. So the question is, how can we learn from that kind of, how are we leveraging this information, which no one really predicted in designing future trials, both when it comes to looking for endpoints, and also like a question to Dr. Shostak with regard of finding targets, because clearly glycemic control was not the right target for predicting or protecting renal outcome. So what should be the targets? What should be the endpoint? What can we take from there to inform future trials?
So Ali, maybe I'll just start with a few comments and then hand over to Catalin. But I'm not sure that it was entirely unexpected. Certainly I was challenged on many occasions while we were running Credence as to why we were doing this study. But we had confidence, and we had confidence based on a few factors. One is that we knew the drug lowered glucose and blood pressure, and that had benefits. The second was that the work of David Cherney and a whole range of others showed important potential mechanisms of glomerular hemodynamic effects. And the third was that in the early data from SGLT2 inhibitors back in 2012, we saw 30 to 40% reductions in albuminuria, and we saw acute effects on EGFR supporting a glomerular hemodynamic effect. So we started Credence in 2013, long before any of the data was out. And we convinced Janssen to invest in a major outcome study, very large numbers of money, and a big commitment from all of us, because we believed that this was going to be beneficial. And fortunately, we happened to be right on this occasion. I think it tells us that we've got to pursue these agents where there are data, particularly that support benefit, and we can get clues from things like albuminuria and EGFR. There's a big debate about what albuminuria means, but I think clearly it means that the drug's having some effect on the kidney that warrants further studies. So I think that's a lesson for us in terms of how we might move forward. And it also tells us we need more albuminuria-like biomarkers that we can use to help us predict the benefit.
I completely agree with you. I think the next challenge, what I see is that, now if we have multiple drugs, how are we gonna align them is there a specific order we will have to prescribe? Is ACE/ARB before SGLT2? If some of the new drugs will be beneficial, are they going to be after all this in sequence, or can we identify patients who are particularly responding for certain class, better for ACE/ARB, especially for SGLT2. But what we are also interested is what are the residual pathways of progression. There is clearly a significant progression for ESRD, even after the use of ACE/ARB and SGLT2 inhibitor. What drives that progression? And then there are lots of new drugs have been developed. There are most of the things, or at least half of the things that are targetable in the genome, there are some tool compounds available. And I think using unbiased data, we can potentially match patients to these new drugs and compounds. And also maybe using these unbiased outcomes, we might be able to see whether these drugs could have an effect on a specific target population, mostly in the context of precision medicine, and using these multi-omics profiling as a readout, an early readout for drug development. So we hope that something like that could be achieved in the context of Trident, so we could actually see what are the residual pathways of progression. Are there specific patients who should be prescribed specific drugs, and then whether we can repurpose existing compounds based on their patient profiles.
Great. So I don't think there are any more questions. This has been a fantastic session. I want to thank again Dr. Sestak and Dr. Perkovich for fantastic talks, and for all the audience that listen attentively and ask great questions. Thank you for joining us, and we'll see you at the next meeting. Thank you.
Thank you so much.
Bye.
Thank you very much. Bye-bye.